Discovering how base pair changes affect protein formation was a huge part of understanding protein synthesis. For example, the difference between a sickle and normal red blood cell is only one nucleotide. However, early DNA scientists had no way of knowing the exact sequence they were studying, only the percent composition. However, in 1975, Nobel laureate Frederick Sanger developed an incredible method for determining this sequence. Let's take a molecular level look. Too fast? Sorry about that. You might be a little bit confused. Why does it look like DNA replication, but weird? Isn't this just PCR? What are those square nucleotides? At a slowed down rate, we can stop and really see what's going on. This reaction is a modified PCR reaction called chain-terminating PCR. This is all happening in a nice solution of other DNA copies, free-floating nucleotides, the oval shapes, DNA replication enzymes, the purple and yellow pieces, and these special molecules called dideoxynucleotide triphosphates, or DDNTPs for short, which are the squares. At first, we only have a double strand of DNA which we do not know the order of, shown as the pink helix. We must split the DNA in two to get individual strands, the target strand and the template strand, which is done by DNA helicase, represented by the yellow rectangle. The target strand is the sequence that we want to know. By using the complementary strand as a template, we can create an exact copy of our target to analyze. Now, a primer and a DNA polymerase attaches to the template strand. The nucleotides are added as normally seen during DNA replication. However, there's a small chance that instead, a DDNTP will be added. These DDNTPs can also contain a fluorescent or radioactive tag for later detection. At this point, the DNA polymerase can't add any more nucleotides due to the chemical makeup of the DDNTP. The remainder of the single strand is unstable and breaks away, leaving a double strand including the DDNTP. Here's the Sanger method again, in real time. As there are many copies of the DNA in this solution, after the reaction they have varying lengths due to the random placement of the DDNTP. This reaction, for example, contains adenine DDNTPs. This means that the terminations will always occur at an A in the sequence. In manual sequencing, this process of adding DDNTPs is repeated 3 more times in separate reactions for the cytosine, guanine, and thymine bases. Then, the strands are analyzed via one of 2 ways. If these reactions were all performed separately, a sample is taken of each the A, T, G, and C, and run through gel electrophoresis. Because the electrophoresis separates the DNA by length, looking at the bands from shortest to longest will indicate the DNA sequence. If the reactions were done together, a sample is taken and run through capillary electrophoresis. Then, the researchers can determine which terminating base pair corresponds to each length by using the different fluorescent markers attached to the DDNTPs. Now, we know the sequence of our target strand. Thank you so much for watching.